Hi, my name is John Liu, and I am an MD PhD student in the laboratory Michael Lonker in the Department of Surgery. And today I'm going to present my talk entitled Meta-analysis of single-cell transcriptomics data after myocardial infarction reveals heterogeneity of cardiac fibroblast response. The motivation for this work is that acute myocardial infarction and ischemic heart disease in general is the leading cause of death and disability, not only in the US but also around the world. And one of the key pathological hallmarks of acute myocardial infarction is the formation of this dense collagen scar. In the short term, it's pretty adaptive because it prevents acute cardiac rupture of the heart, but over the long term, it becomes maladaptive and it triggers hypertrophy and fibrosis in the remaining myocardium, leading to ischemic cardiomyopathy. So in this present study, we wanted to understand how exactly does this scar form, how does it remodel over time, and what are the pathways that sort of activate the formation of this collagen scar, in particular, the formation of the scar by cardiac fibroblasts, which lay down the scar tissue in the heart. So in particular, we sought to leverage the recent proliferation of single-cell transcriptomic studies that looked at a small number of time points after myocardial infarction, but because different studies looked at different time points, in the study, we wanted to create a meta-analysis to look at all those studies, all those different time points after myocardial infarction to come to a comprehensive conclusion. So in terms of our methods, we generated a comprehensive atlas of mouse cardiac fibroblasts after myocardial infarction by taking in data from 9 different studies that in total encompass 54 different mouse samples after myocardial infarction. And in particular, our 54 samples included 9 healthy samples, 12 sham samples, and then 33 samples after myocardial infarction at different time points after myocardial infarction. And in panel A of figure 1, we have the UMAP plot that now plots all 131,000 cardiac fibroblasts in our comprehensive atlas of cardiac fibroblasts. And now each cell is represented by a single dot. And these dots, these cells are clustered based on their relative gene expression of all 20,000 genes in the human genome. And what we can immediately appreciate is that there's a wide heterogeneity of cardiac fibroblasts, and these different clusters of subtypes of cardiac fibroblasts are colored in different colors, and they all express different canonical marker genes. And we can also appreciate that all the cardiac fibroblasts that we've annotated also express universal cardiac fibroblast markers like DPT or PDGFR alpha. We then asked, how exactly do the different subpopulations of cardiac fibroblasts, how do they change at different time points after myocardial infarction? And then how do they change at different time points after a sham surgery? And when we plot that, we can immediately appreciate that only one subtype of cardiac fibroblasts, these subtype colored in red, or these myofibroblasts, preferentially expand after myocardial infarction, peaking at days around 5 through 14. But a similar sort of pattern is not observed in the sham-operated mice. And we can quantify that in panel D. And we show that at baseline, 15% of all cardiac fibroblasts are myofibroblasts, but at days 5 through 17, it becomes up to 50%, 30% of cardiac fibroblasts become myofibroblasts. And what we can further show is that at these time points after myocardial infarction, that the production of these extracellular matrix proteins, including collagens type 1 and 3, they also peak around days 5 through 14. We next asked the question of what exactly is causing these myofibroblasts to have this dramatic proliferation after myocardial infarction. And the way we approached this was by performing gene ontology analysis of the top marker genes of the myofibroblast subcluster. And what we observe are that a lot of pathways related to sensing mechanical tension are actually upregulated in these myofibroblasts, including the YAP-TAZ pathway and also FAK signaling pathways. And what we can also appreciate in panels G through H is that the expression of these different mechanosensory proteins, including PTK or focal adhesion kinase and YAP, are both universally expressed across all cardiac fibroblasts, and also these piezo-1 and piezo-2 mechanosensory ion channels are also widely expressed across all cardiac fibroblasts. And in particular, piezo-2 is exclusively expressed in the myofibroblasts, which is a pretty interesting sort of phenotype. And then we asked the question of why exactly are mechanosensation related to the activation of cardiac fibroblasts. And then we recalled in panel A of figure 2 that the law of Laplace states that the wall stress or the mechanical tension experienced on the ventricular free wall is inversely related to the thickness of that wall. And what we can appreciate immediately from the histology of heart samples after myocardial infarction is that the wall thickness of the left ventricular free wall immediately drops after a myocardial infarction, even before the onset of fibrosis as quantified in panel C through D. So for instance, in day 3 after MI, there's decreased wall thickness, but no fibrosis, which may suggest a potential pathway for how increased mechanical tension through this increased wall stress, as predicted by the law of Laplace, actually then activates these mechanosensory genes that we show to be upregulated through our single-cell meta-analysis. So in conclusion, we find that cardiac fibroblasts exhibit significant heterogeneity in response to myocardial infarction, and that in particular, only one subtype of cardiac fibroblasts, these myofibroblasts, preferentially expand after myocardial infarction, and that these myofibroblasts also express a lot of these extracellular matrix proteins. And then further, we show that these myofibroblasts also express a bunch of mechanosensory genes, which provides a potential pathway for cardiac fibroblast activation. In the future, we want to functionally validate these mechanosensory genes. Thank you for your time. Bye-bye.